This is the Illuminate recorded lecture for Chapter 20, Anatomy and Physiology, Semester 2. This will be about the immune system. We're going to talk about innate and adaptive body defenses. We're going to walk through the immune system from front to back and talk about antibodies, which would be a humoral response, and then we'll talk about T cells and the cellular response, phagocytosis, and how that all works together to create an immune system for us. Immunity is really defined as the resistance to disease, our ability to protect ourselves against bacteria, viruses, or any other kind of microorganism or larger organism that would harm us. The immune system has two intrinsic systems, one which is called an innate system, one is what's called an adaptive system. The innate system is nonspecific. In other words, it's a defense system that doesn't have it's not antigen specific when it responds to something. It does the same response no matter what kind of antigen or what kind of invader comes into our body. An adaptive defense system is more specific. It's where we develop very specific antigen-based defenses or very specific defense mechanisms for a given invader. And so it's kind of the difference of an innate a defense system would be more like when you have a fever, when you get a cold. You have a fever, whether you're any kind of infection, and you get a fever, you get a different kind of fever for a different kind of infection. Adaptive would be more like developing tetanus antibodies when you get a tetanus shot. When you get a tetanus vaccination, you don't develop antibodies to everything. You just develop antibodies to the tetanus vaccine or the tetanus toxoid. And so that, if you will, is more specific. That's just the differences in the two. We talk about the innate and adaptive system here. We'll talk about innate defense systems has two lines of defense. We have a first and a second line of defense. The first line of defense is that external body membranes, the skin and the mucosa, and all the openings into our body. And then we have a second line of defense, and that's really antimicrobial proteins, phagocytes, which eat the invader, and other types of cells which inhibit the spread of the invaders. And of the second line of defense, inflammation is the most important mechanism. We're going to talk about that to an extreme in just a bit. The other type of immunity is the adaptive defense immunity we talked about. It's the third line of defense. And it attacks foreign substances we call antigens at a very specific rate. It takes longer to react than the innate system, which is available at all times. The adaptive system has to go through a process, and sometimes a little slower. So innate and adaptive defenses, they're deeply intertwined. We have they interrelate quite a bit. And this talks about the innate and adaptive senses. Innate senses, you'll see, include the surface barriers. That would be the skin and the mucous membranes. Also the internal defenses, the phagocytes, those white cells that eat other cells or eat other bacteria or viruses. NK cells, which are killer cells. Inflammation, which is a general defense process. Antimicrobial proteins, which react the same way to any bacteria or virus. And then, of course, fever, which is very nonspecific. Where adaptive defenses have a humoral and a cellular response. Humoral refers to B cells, and that's B cells produce antibodies. So antibody response is a humoral response. T cells are cells that don't produce antibodies, but they create a cellular immunity. We'll talk a lot more about T and B cells in later slides. We talk about innate defenses of the skin barriers. We're really talking about the skin, the mucous membranes, and basically the secretions. Mucus secretions, the tears from the eye, if you will, and they create a physical barrier to most microorganisms. Keratin, that substance that's in the white that are in your cells of your skin, surface area of your skin, is resistant to weak acids and bases, a lot of bacterial enzymes and toxins, so it gives us protection. And finally, mucosa, that's the lining, if you will, to every orifice that enters the body, provides a similar mechanical barrier. It sort of traps the bacteria in its mucus, if you will, and it sort of takes care of that. Surface barriers have protective chemicals, which may inhibit or destroy microorganisms, and they include skin acidity, or the acid content of the skin, our lipids in the sebum and dermacidin in the sweat, hydrochloric acid and protein digestive enzymes in the stomach mucosa, and then lysozymes of the saliva and lacrimal fluid, and finally mucus, which is just a trapping mechanism. We also have respiratory system modifications. We have mucus-coated hairs in our nose and our oral and nasal mucosa, as well as the bronchioles going all the way down to the alveoli. The alveoli do not have mucus lining, if you will, but everything else does. The cilia of the upper respiratory tract sweep the dust and bacteria-laden mucus to the lower respiratory tract and the passages, and we either cough it up or swallow it. Internal defenses include cells and chemicals, and these are still within those general defenses. They're necessary if microorganisms actually are able to invade into our deeper tissues. And we talked about those. Phagocytes, those are white blood cells that eat other bacteria or cells or whatever. We have NK cells, which are those natural killer cells. We have the inflammatory response where we have macrophages and white blood cells and mast cells and inflammatory chemicals, and we'll talk about a lot about those in a bit. And then we have antimicrobial proteins like interferons and complement proteins, which work the same on any bacteria or virus. They're very general. And of course, we said fever before, and so that's the second time we've said that. We're talking about phagocytes. We're really talking about macrophages and neutrophils. Macrophages develop from monocytes, a type of white blood cell, to become the chief phagocytic cell. Free macrophages wander through tissue spaces. They wander all the way through like freewheeling cells. For instance, alveolar macrophages live in the alveoli of the pulmonary area. Fixed macrophages are permanent residents of some organs. For instance, we have Kupffer cells, which are permanent macrophages of the liver. We have microglia, which are those permanent glia cells of the brain. And then we have another macrophage called, another phagocytic cell called the neutrophil, and it becomes phagocytic when it encounters infectious material or tissue. So these two really make up all the phagocytic cells of the body. The macrophages, which actually originate from monocytes, and the neutrophils, which are a type of white blood cell also. The mechanism of phagocytosis is that the adherence to phagocytes to the pathogen. In other words, the phagocyte, if you will, the cell, comes in contact with a pathogen, and it's facilitated by optimization. Opsonization is really where the pathogen gets coated by these complement proteins, we'll talk more about later, or antibodies, and it sort of alerts the phagocyte to attack that particular cell or that foreign tissue. And we're going to talk more about that later, but for right now I just want you to get the idea of how the phagocytic process works. Phagocytes are either macrophages or neutrophils. They're facilitated, or they are able to see the material they're supposed to phagocytize based on what may be called opsonization. That's where it's a coating or an attachment, if you will, around the pathogen by either complement proteins or antibodies, and we'll talk about that again a little later. This is an electron microscopy of a phagocyte, a macrophage, you will, using its cytoplasmic extensions to pull the bacteria towards itself. You see the bacteria in these little green spherical forms, and you see that macrophage, and that's really what it looks like. This is an actual picture of that process. So let's talk about the events of phagocytosis. Let's talk about that process that the phagocytic cell has to go through to actually commit phagocytosis. Let's see what this says. So here the phagocyte adheres to pathogen debris, and so you see those pathogens there, and it essentially encircles that pathogen, and through endocytosis brings it into the cell. So the phagocyte forms pseudopods that eventually engulf the particle and form that phagosome, that encapsulated little bag. Lysosomes in the cell fuse with that bag and start to break that down, forming a phagolysosome. And really, what happens is this starts to get eaten up. The lysosome enzymes digest the particles, leaving a residual body, if you will, and then the cell then extrudes the material that's left over, that's exocytosis, out and removes that indigestible and residual material. So it actually uses this for nutrients to some degree, but mostly it just processes it, gets it inside. The lysosomes, the digestive enzymes kill it, they destroy what they can destroy, it absorbs what it can absorb, and finally extrudes whatever remnants there is of that bacteria, that foreign material. This talks about the mechanism again, destruction of pathogens. There's acidification, digestion by the lysosomal enzymes. We already talked about that. There's a respiration burst. That means there's a release of cell-killing free radicals, an activation of additional enzymes. And then there's oxidizing chemicals. There's H2O2 and the defensins, if you will, in neutrophils. So those are other mechanisms that go along with that phagocytosis process. We talked about those NK cells, natural killer cells, and those are really large granular lymphocytes. The target cells that lack self-surface receptors. In other words, they don't really they kill all bacteria or viruses the same. They're not antigen or invader specific. So they don't have any self-surface receptors that allow them to make those changes. They induce what's that apoptosis in cancer cells. We'll talk about what that means later, and virus-infected cells. And so they induce that process, and they secrete a potent chemical that really enhances or creates that inflammatory response. So they help to reduce the inflammatory response. What I want you to leave with: natural killer cells, NK cells, are large granular lymphocytes. They don't have any cell surface receptors or self-surface receptors. They induce apoptosis of the cancer cells and virus-infected cells, and they secrete chemicals that really help to move along the inflammatory response. The inflammatory response is triggered whenever body tissues are injured or infected. It helps to prevent the spread of damaging agents throughout the body, and it helps to dispose of cell debris and pathogens. And it also sets the stage for repair. We were talking about the skin. We talked about the inflammatory responses, so this should be kind of a review. The inflammatory response: the cardinal signs of acute inflammation include redness, heat, swelling, pain, and sometimes immobilization or impairment of function where the joint can't move because of the swelling, if you will. The inflammatory response to macrophages we talked about, and the epithelial cells, the boundary tissues, they're what are called toll-like receptors, TLRs. The TLRs recognize specific classes of infecting microbes. And activated TLRs trigger the release of what are called cytokines. Cyto means cells, kines mean killers, that promote inflammation. So that's another step to promoting inflammation. The inflammatory mediators include chemicals that help to create that, if you will, or create the inflammatory response, are histamine, and those mast cells actually contain little granules of histamine. Blood proteins, those proteins in the blood that sort of perpetuate that. Kinins, which are, and prostaglandins, PGs, leukotrienes, and complement, which help to perpetuate the inflammatory response. And those are released by injured tissue, by the phagocytes we talked about, including macrophages and neutrophils, the lymphocytes we talked about, basophils, and mast cells. So you kind of get an idea what where we're at now. Vasodilation and increased vascular permeability actually is a step in the inflammatory process. It allows inflammatory chemicals which cause dilation of arterioles, resulting in what's called hyperemia, or an excess of blood in the area, more circulation. There's increased permeability of the local capillaries and edema, and that edema leaks exudate outside of the capillary into the surrounding tissue. And that exudate contains protein and clotting factors and antibodies, as well as some of the white cells that are going to attack that area. Edema is really functions of the surge of the exudate, the swelling, because the exudate has left the cardiovascular system, the capillaries, and moves the foreign material into the lymphocytic vessels. It delivers clotting proteins to form a scaffold, or a skeleton, if you will, for repair, and to isolate the area so that the infection or inflammation can't move any farther out. And this is a very long diagram which talks about tissue repair, and it's worthwhile to go through it. So there's tissue injury, there's release of chemical mediators, histamine, complement, kinins, prostaglandins, that cause vasodilatation of the arterioles and capillaries, increased capillary permeability, and which attracts the neutrophils, monocytes, and lymphocytes to the area. Now, also what happens if you follow the vasodilatation is there's local hyperemia, increased blood flow to the area, which causes heat and redness to the area, and that locally increases the temperature, which is why areas where we have an infection get a little warmer. You see that increased permeability also causes the capillaries to leak fluid. That leak fluid is rich in protein. It causes pain and swelling, and there's a possible joint limitation here when that happens, but those are the four cardinal signs. you see that when we attract neutrophils, we move right over to this other area, and we'll talk more about that. But the other thing that happens with a capillary leak is that you get leak clotting proteins and temporary fibrin patch, that scaffolding we talked about. We have a release of leukocytosis-induced factor, which causes leuko, meaning white, cyto, meaning cell, and a condition of. Those white cells, we create, if you will, increased numbers of white blood cells go in the bloodstream. They migrate to the injured area. They go through marginalization. That means they are able to escape through the walls of the capillary. They have diapetesis, which is leukocytes pass through the cell walls. Phagocytosis of pathogens and dead tissue occurs by the neutrophils in the short term, by macrophiles in the long term, and they are cleared of that debris. And so that's the process we go through at the site, if you will. Phagocytic mobilization includes the neutrophils, then phagocytes flood to the inflamed area. The steps for phagocyte mobilization include leukocytosis. That's an increase of white blood cells, which release of neutrophils from the bone marrow in response to leukocytosis-inducing factors or chemicals that are released by the injured cells. There's marginalization. That means the neutrophils cling to the wall of the capillaries in the inflamed area. And then there's diapetesis, as we talked about, in neutrophils, where they escape through small openings in the capillaries, which allow them to directly attack whatever's foreign to our body. And the chemotaxis, where the inflammatory chemicals, chemotactic agents, promote positive chemotaxis of the neutrophils. So let's look at this diagram of how the inflammatory response actually occurs. We have an injury. In this case, you see this nail sticking through the skin. Inflammatory chemicals diffuse from the inflamed site and act as what are called chemotactic agents, if you will. That causes leukocytosis. Neutrophils enter blood from the bone marrow, and so you have those in the capillaries. The marginalization occurs. That's where the neutrophils cling to the wall of the capillary. Diapetesis occurs, and that's where they ooze through or flatten themselves and go through small openings within the capillary wall. And finally, chemotaxis, where the neutrophils follow the chemical trail left by the tissue injury and start to attack the injured area directly. Now, antimicrobial proteins are also part of our inflammatory response, and they include primarily interferons, IFNs, and complement proteins. Interferons and complement, if you will, attacks microorganisms directly. Again, this is a general response, not a specific response. And their job is to hinder the microorganism's ability to reproduce, and they also will sort of mark the organism so that other bacteria or other phagocytic cells can see that material, if you will, that antigen, and know that it's supposed to be in the body and it'll attack it directly. For instance, viral infected cells are activated to secrete interferons, which give us some protection. Interferons enter those neighboring cells, so those viral infected cells, if you will, activate and create IFNs, interferons, that attack the neighboring, or that enter the neighboring cells, and so the neighboring cells produce those antiviral proteins that help to block viral production. Now, it isn't a great system. It doesn't work on everything because viruses are smarter and faster than our ability to, or interferons' ability to respond to that. But nevertheless, it's a means that allows us some immunity from our innate response. In this graphic, we can see how that virus works, or how that interferon is created. The virus enters the cell, and so there's a nucleic acid that goes with that. The virus enters the cell. What you're seeing then is the cell then produces a interferon, and as it's being dealt with by the virus, it releases that interferon, which goes to other cells and provides some protection, if you will, provides protective protein for some of those cells. It's not a great system, but it's a system. You see the interferon genes here, they switch the DNA, making that cell produce more of that virus. You see that as that happens, the cell produces that interferon molecules, which leave the cell. And finally, that interferon binding stimulates cells to turn on genes for antiviral proteins. So it kind of causes the other cells to do what they're supposed to do and has some protection. Again, it's not a great system. And finally, antiviral proteins block the viral reproduction, and this last step. Interferons are produced by a variety of body cells. Lymphocytes produce a gamma or immune interferon, where most other white blood cells produce an alpha interferon. Fibroblasts produce what's called a beta interferon, and interferons are also activate macrophages, and they mobilize those natural killing cells, if you will, to attack the invader. So interferon does a lot of things. Its functions: it's antiviral. It reduces inflammation. It activates the macrophages and mobilizes the natural killing cells. The interferon is genetically engineered, can be genetically engineered, and we use it as antiviral agents against hepatitis and genital warts, and also multiple sclerosis treatment. So we've engineered or artificially created interferon, which we use against other agents now. And so you're going to see some of that. Interferon is also an important treatment for HIV. The other thing we talked about, as far as a protein is released, is complement. In complement, this is there 20 blood proteins that circulate in an inactive form. I think the number is actually closer to 30, the last time I checked through the New England Journal of Medicine. But it includes complement factors C1 through C9, factors B, D, and P, and some regulatory proteins. And we're not going to go into these too closely, but I just kind of want you to know that those are important factors we're going to talk about. And they are a major mechanism for destroying foreign substances. They have a very specific action. We'll talk about how that works. Complement amplifies all the aspects of the inflammatory response. It makes it better. It's synergistic to the other parts of the response. It kills bacteria and certain other cell types by cell lysis. It helps to destroy the cell. It enhances both nonspecific and specific defenses. We're talking about nonspecific in this area, but when we get to specific, complement will also play an important part. Excuse me. There are two pathways where complement is activated. There's the classical pathway and what's called an alternative pathway. The classical pathway is where antibodies bind to the invading organisms. Complement 1 binds to the antigen-antibody complex, that is, the antibodies have attached to the antigen, that's that foreign substance, and then complement then binds to that, creating what's called a complement fixation complex. There's an alternative pathway, which is triggered when when it's activated, C3, B, D, and P interact on the surface of the microorganism. We're going to talk about both those pathways in just a moment. And into the complement activation, each pathway involves a very specific orderly sequence of activation proteins to allow them to be successful. And each step, if you will, is necessary for the next one. So it says here each step catalyzes the next. What it's really trying to say is that each step is necessary in this progression, this cascade, if you will. Both pathways converge at the complement C3, which cleaves into C3A and B to cause that action to occur. And I'm going to show you that in just a minute. Activated complement enhances the inflammatory process, promotes phagocytosis by the mast cells and the neutrophils, causes cell lysis, where C3b initiates the formation of what's called a membrane attack complex, or a MAC. The MAC causes cell lysis by inducing a massive influx of water. It actually drills holes through the cell wall, if you will. C3b also causes opsonization, which is basically coating the cell or the bacteria or the foreign body with complement so that phagocytes can see it as foreign. And C3A, if you will, causes an inflammation. So it all kind of works in this little cascade. You see here that the classic pathway, you have an antigen-antibody complex which initiates C1, C4, and C2, and that causes this cascade. The ultimate end here is that there's a hole, if you will, drilled through the cell membrane of that particular bacteria, and that it allows the efflux of the interior of the cell to go outside the cell, so the cell dies, obviously. In the alternative pathway, there's spontaneous activation, there's stabilizing factors B, D, and P. No inhibitors on the pathogen surface, enhances inflammation, which stimulates histamine release, increases blood vessel permeability, attracts phagocytosis by chemotaxis, and so it works a little differently. But you get the idea of how those two work together through that process. The next symptom of an inflammation or inflammatory response is, of course, fever. And again, it's a general response. It's not a specific response. It's a systemic response to invade organisms. Your whole body goes into a fever, not just part of your body. Leukocytes, white blood cells, and macrophages, those monocytes that become phagocytic, are exposed to foreign substances which create what are called pyrogens. And pyrogens sort of reset the body's thermostat upward so we develop a higher temperature. What you're going to see is you'll see high fevers, which could be dangerous because heat obviously denatures the enzymes, and so if you take our temperature up too high, most of our body processes stop working very well. But the benefits of a moderate fever are it causes the liver and spleen to sequester iron and zinc, which is really what the microorganisms need to replicate. So it's sort of sequester means to hold back or to secret away those minerals of iron and zinc. It also increases our metabolic rate, which speeds up our repair, more blood flow, that kind of thing. We have adaptive defenses also. We talked about before inherent defenses, but adaptive defenses, which are immune-specific defenses. This is where the body creates an actual response, a smart bomb, if you will, that attacks a very specific bacteria or antigen. It protects against infecting agents and abnormal body cells, and it amplifies the inflammatory response because it's much more specific. It also activates complement. We talked about that before. Adaptive immune response is, like I said, specific. It's systemic. It affects the whole body, and it has a memory. That means that once we've reacted to a certain kind of antigen, then the body's ability to react to it in the future is much better because it has some memory that it has happened before. How does that work? Well, if you get a tetanus shot, what happens is the tetanus immunization causes your body to produce, guess what? Certain cells, B cells, we'll talk about that, and some of those cells attack the virus or the bacteria that we put into you. We have the tetanus vaccination, and some of those cells become memory cells. So they hide themselves away. So if tetanus ever shows up again, then we can react rather quickly to that. Now that's what an immunization is all about. It helps us to react faster to the next event, and I'll talk about that in just a bit. There are two separate overlapping arms, if you will, or layers of adaptive defense. There's a humoral, which is antibody mediated, and there's a cellular response, which is cellular mediated. The antibody mediated response is primarily B lymphocytes. B as in boy, lymphocytes. And the cellular response is phagocytes. We talked about macrophages and neutrophils. But as far as the specific part of that, that would be T cells, T as in Tim. And we'll talk about each one of those as we go through the next few slides. Remember that antigens are substances that can mobilize the adaptive defenses and provoke an immune response. An antigen is anything, if you will, that identifies a cell as foreign. We have antigens on our cells. We do. Every one of your cells has a stamp on it with several antigens that identify each of those cells as yours. But when something foreign comes into your body is able to identify the surface areas of those cells, their antigens, if you will, and be able to create a response, a defense response. Most are large and complex molecules, and they're not normally found in the body, so they're what are called non-self or non-id self. ID comes from, the id response means your own response, sort of Freudian, if you will. But so you do have antigens on your own cells, but they are self antigens. That means they belong to you and the body knows that. But when something foreign comes in, it's a non-self antigen, meaning that it's a non-id antigen, which means the body sees that as foreign. Now, antigens come in a couple flavors. We have complete antigens, and their important functional properties include immunogenicity, that is the ability to stimulate proliferation of specific lymphocytes and antibodies. So that complete antigen it causes our body to send out specific lymphocytes and create specific antibodies to fight that antigen. They also have reactivity, which is the ability to react with products of activated lymphocytes and antibodies released. So there's important functional properties. So examples are foreign proteins, polysaccharides, lipids, and nucleic acids. These are all foreign, if you will, elements. Haptens, which is another name for an incomplete antigen, are much smaller molecules, which include peptides, nucleotides, and hormones. They're not immunogenic by themselves, but they are immunogenic when they're attached to body proteins. and they cause the immune system to mount kind of a harmful attack. For instance, some of those include poison ivy, where we react to the poison, the content of the poison, the resin of the poison ivy. Animal dander, where we have a reaction to animals, to cats, and allergies to cats, dogs, horses, whatever. Detergents, where some people have a high sensitivity to certain detergents, and certain cosmetics where people will react to that. That is still a reaction, but the molecules are much smaller, the way it reacts is much different, quite a bit different, if you will, than that complete antigen reaction we were talking about previously. Antigenic determinants, certain parts of an entire antigen that are immunogenic. That's what we're talking about. Antibodies and lymphocyte receptors bind to them, and most naturally occurring antigens have numerous antigenic determinants that mobilize several different lymphocytic populations, form different kinds of antibodies, if you will. They're large, they're chemically simple molecules like plastics. They have little or no immunogenicity. So what we're talking about here is antigenic determinants, how that occurs, and as we go through that. This is an antigen that something that would, in this case, it says forms the body, and we have an antibody A, B, and C here, and you see that the antibodies can only attach very specifically to a specific receptor on that antigen, and this talks about the antigenic determinants. So this is a very simplistic way of looking at it, but only certain antibodies can attach to certain antigens. So if you have an A antibody, it can only attach to an A antigen, or a B antibody can only attach to a B antigen, as it were. Antibodies are what we produce. Antigens are the stuff that's on the surface of cells and specifically bacteria or anything foreign. We have self-antigens, and those self-antigens are MHC proteins. They're protein molecules, self-antigens, which are on the surface of our cells. They're antigenic to others, but not to us. So you have your own MHC proteins that make up your self-antigens on each of your cells. And it's sort of like stamped. Every cell you have is stamped with these self-antigens. Now, if you take your blood and you put it into somebody else, then it would be antigenic to them because your proteins would be foreign to them, just as their proteins would be foreign to you if they gave you their blood and it wasn't done in a specific way. MHC proteins are coded for by genes of the major histocompatibility complex, MHC, and are unique to an individual. So MHC stands for major histocompatibility complex, MHC antigens. That means that there are antigens. They belong to you, IDRI antigens, self-antigens. MHC proteins, there are classes, which include class 1, which is found in virtually all body cells, and class 2, which are found on certain cells in our immune response in our immune system. MHC proteins display peptides, usually self-antigens, and in infected cells, MHC proteins display fragments of foreign antigens, which helps to mobilize our immune response. So against those cells. So our body says, not a normal, those antigens are normal, and we need to attack that. So if you have a cell that's defective, or a cell that is infected, or a cell that is dying, then the MHC fragments will start to show, and it will look foreign to our body, and our body will mobilize against it. The cells of the adaptive immune system include two types of lymphocytes and what are called APCs, or antigen-presenting cells. The most important we'll talk about are really the lymphocytes. And there are two kinds of lymphocytes, as I said. There's B lymphocytes, or B cells, which are which provide humoral immunity, that is antibody-generated immunity. Then we have T lymphocytes, T cells, which provide cell-mediated immunity. So they do very different things. And we'll talk about how they develop in just a little bit. You also have antigen-presenting cells, APCs, which don't really respond to specific antigens, and they play essentially an auxiliary role in our immunity system. But they are present, and we need to just mention them. All lymphocytes originate in the red bone marrow. B cells mature in the red bone marrow, and T cells, if you will, aggregate or move to the thymus gland, where they mature there, i.e. T lymphocytes, thymus gland. When they mature, they have immunocompetence. That means they are able to recognize and bind to specific antigens. They have self-tolerance, which means they are unresponsive to self-antigens, so they can bind to specific antigens, but they are unresponsive to our id antigens, our self-antigens. NAVE, which is a term we use for unexposed B and T cells, are exported in that NAVE form to lymph nodes, spleen, and other lymphoid glands, so that if they ever, so they're out there, so they're sort of deployed, if you will, to those areas. And so if they come in contact with an antigen, then they'll have a reaction. They'll become less NAVE. They'll become activated, as a word. So it just shows that the immature lymphocytes are located in the bone marrow. Some go to the thymus gland, and they are sent off, if you will, or activated, if you want to activate, excuse me, they're deployed to lymph nodes, spleen, and other tissues, and throughout the bloodstream. And you can see that the B lymphocytes, those are T lymphocytes, B lymphocytes are staying in the bone marrow, but are also deployed to spleen in the same places as the T lymphocytes. So lymphocytes are destined to become T, migrate to the blood, the thymus, and develop that immunocompetence there. B cells develop immunocompetence in the red bone marrow. Immunocompetent but still naive lymphocytes, it's unactivated lymphocytes, leave the thymus and bone marrow, and they seed, if you will, or are deployed throughout the lymph nodes, the spleen, and other lymphoid tissues when they encounter their antigens. Antigen-activated immunocompetent lymphocytes, those are activated lymphocytes, effector cells and memory cells, circulate continuously in the bloodstream and the lymph and throughout the lymphoid organs of the body. And we'll talk a lot about how that's going to happen in just a bit. T cells mature in the thymus, as we said, under what's called a negative and positive selection pressure. The positive selection is that it selects the positive selection selects T cells capable of binding to self-MHC proteins or antigens. The negative selection prompts apoptosis of the T cells that bind to self-antigens displayed by that self-MHC. So that what they're really looking for is it kind of ensures self-tolerance is what we're trying to do. And so those are the two reactions we have with T cells. You see that positive selection, if you will, T cells will recognize their self-major histocompatibility proteins of our MHC, and you see failure to recognize the self-MHC results in apoptosis or death of that cell by suicide. Recognizing the self-MHC results in MHC restriction. That is, survivors are restricted to recognize antigens on the self-MHC. Survivors proceed to negative selection. So negative selection, the T cells must not recognize self-antigens. The recognizing self-antigen results in apoptosis. So it's exactly the opposite. One reacts if it doesn't. If you want, one reacts in one direction, one reacts in the opposite direction, i.e., the positive and the negative reaction. Failure to recognize that cell by entirely the self-antigen results in survival and continued maturation. So they work in a couple of different ways. Bone cells, which are in the bone marrow, and they're self-reactive B cells, which are eliminated by apoptosis, clonal deletion, or undergo receptor editing. That's the rearrangement of the receptors. They're inactivated, it's called anergy, if they escape from the bone marrow. So that's just a way of managing things in a normal way. Antigen receptor diversity. Lymphocytes make up to 1 billion different types of antigen receptors. They're coded for 25,000 or better genes, and the gene segments are shuffled by what's called semantic recombination. Genes determine which foreign substances the immune system will recognize and resist. So that's the diversity of our lymphocytes. That's pretty cool when you consider they're coded for 25,000 different genes. And the gene segments are shuffled by somatic recombination, and they help determine how we're going to react to a foreign substance or a foreign antigen. Antigen-presenting cells, APCs, engulf the antigens. They present fragments of antigens to be recognized by the T cells. There are major types of APCs: dendritic cells in the connective tissue and epidermis, macrophages in the connective tissue and lymphoid cells, and the B cells we talked about earlier. This is actually a picture of if you will, of an APC, which is kind of interesting. I just want you to look at that. It looks very Star Wars-y, doesn't it? Macrophages and dendritic cells present antigens that activate T cells. The macrophages mostly remain fixed in the lymphoid organs. The dendritic cells internalize pathogens, and interlymphatics present the antigens to the T cells in the lymphoid glands. So the dendritic cells engulf, if you will, or internalize the pathogens, and they present them to the T cells, the lymphocytes. The activated T cells come in contact with the antigen, and the activated T cells release chemicals right away that prod the macrophages to become insatiable phagocytes and to secrete bacterial cytochemicals. So what happens is the antigens activate T cells, is how it works. Macrophages mostly remain fixed in lymphoid tissue. The dendritic cells internalize those pathogens, interlymphatics, and present the antigens to the T cells, which now become activated. The activated T cells release those chemicals that sort of make the macrophages hyper and become just totally insatiable to phagocytize and secrete bacterial chemicals because they realize that there's a foreign body present. The adaptive immunity, the summary, uses lymphocytes, APCs, and specific molecules to identify and destroy non-self or non-id substances. Those are substances that are not part of our own body. And that depends upon the ability of cells to recognize antigens by binding to them and to communicate with one another so that the whole system mounts an organized response. We talk about different kind of response. We'll talk about humoral immunity response. Humoral is basically an antibody response that's initiated through the B cell system. Antigen challenge is the first encounter between an antigen. So the first time our body sees an antigen, it comes in contact with a B cell, and it comes in contact with what's called an immunocompetent B cell, but it's an A B cell or inactive. That usually occurs in the spleen of the lymph node. If the lymphocyte is a B cell, the antigen provokes a humoral immune response, and antibodies are then produced by that B cell. But then the B cell is activated when the antigens bind to its surface receptors and it cross-links them. The receptor-mediated endocytosis of cross-linked antigen receptor complexes occurs, and the stimulated B cells grow to form a clone of identical cells bearing the same antigen-specific receptors. So they've already been activated. These other ones are already activated. And the T cells are usually required to help the B cells achieve that full activation. So B cells are activated with antigens bind to them. They then clone. They start to produce more B cells, which have a very specific set of antibodies that can attack that specific antigen. The fate of clones, most clone cells become what are called plasma cells, which is the same thing. They're plasma cells that produce antibodies that fight that particular infection and the initial presentation. They secrete the specific antibodies at a rate of about 2,000 molecules per second for 4 or 5 days. The secrete antibodies circulate in blood or lymph and bind to those free antigens that invade her. They mark the antigens for destruction. Clone cells that do not become plasma cells sort of go and hibernate and become what are called memory cells. And they are memory cells that provide the immunological memory, so that the next time that antigen shows up, instead of waiting to develop these activated cells, you already have them. And it shortens that circuit a little bit, that time frame. They mount an immediate response to future exposures of the same antigen. That's exactly how vaccinations work. You get a vaccination, hopefully it's the first time you've ever seen that particular antigen. Your body mounts a reaction to it, kills it off very nicely because vaccinations aren't meant to kill you. And then what happens is not so much that it takes care of the antigens in the vaccination, but it also stores or causes these memory cells to hibernate. So the next time that you see that particular antigen, your body reacts to it very quickly. There's no delay. And for instance, tetanus shots. It's better that if you let tetanus go on, it will kill you. But if you have tetanus shots, what happens is your body already has those memory cells, if you will, in the background. So when tetanus shows up, it's able to fight that rather quickly. This is just kind of an example of how that works. You see the primary encounter, the initial encounter, the antigen comes in contact with those naive B cells, and those B cells then are activated and they start to proliferate. They mostly proliferate in more activated B cells, which are called plasma cells. And they contain that antibody to fight that specific antigen. A few of the cells become what are called memory cells, and they are hibernating cells that go off and wait for the antigen to show up again. So this continues the process on through until the infection or the foreign antigen is destroyed. So let's talk a little bit about immunological memory. There's a primary immune response and a secondary immune response. The first primary immune response is our first contact with that foreign antigen, that specific antigen. Unfortunately, when we come in contact with that antigen, our body activates in its specific way. There's a lag period of 3 to 6 days. And so peak levels of the plasma antibody against that bacteria or that antigen isn't reached for 10 days, and the antibody levels have declined. So there's a long haul there where our body is really subjected to those bacteria or those antigens before the body responds. In the secondary immune response, that is where the body sees the antigen for the second time, and this is what occurs when there's a re-exposure, those sensitized memory cells respond within hours. Instead of waiting for 10 days, they respond in hours. Antibody levels peak in 2 or 3 days at much higher levels, and antibodies bind with greater affinity, and the antibody levels can remain high for weeks or months. So there's great value in immunizations when you think about what happens there. You end up with that high level in the secondary response versus the primary response. This kind of looks at that. There's that primary response. There's the antigen binding to the receptor-specific B cells. B cells with non-complementary receptors remain. There's the memory cell. We've already done all that. But the memory cell, which has been hibernating, now comes in contact with that bacteria again. And within hours, it starts producing lots and lots of plasma cells that are very specific. So to get to this point takes hours instead of days. 10 days, we're talking hours. And then again, there's some memory cells that are reproduced again for the next encounter, the tertiary response or the quaternary response or whatever works. And this kind of gives you a nice little slide about that. The first exposure here is 0. You can see the antibodies start to react, and within 28 days they go away, but it takes a good 10 days before they hit their peak. Where you get the secondary exposure, we've already had those memory cells available, and guess what? The antibody reaction is greater, far greater, goes from like 10 to the 1 to 10 to the 4th, and you start to see a very long tail, as it were, so it's drawn out. Active humoral immunity occurs when B cells encounter antigens and produce specific antibodies against them. There are two, and we're going to talk a little bit about this. There are two types of active humoral immunities. There's naturally acquired, and there's artificially acquired. Let me help you separate those. Naturally acquired, there's no needle involved. Artificially acquired, there is a needle involved. And the thing that's important to notice is that naturally acquired means we had to get the infection. So if we have active humoral immunity, that means naturally occurring, we had to get the infection. So to produce antibodies, to produce those memory cells, we have to have the infection. Or we have to be injected with something that creates sort of an attenuated or a small level of infection, so we produce antibodies against it. So active humoral immunity means antibodies are created. Our body is actively involved. And so it can be naturally occurring when we get an infection, or artificially occurring where we inject somebody with a needle with some antigens, like in tetanus immunizations, where we're giving them dead or attenuated pathogens, which will help pretend like there's an infection, and the body will, again, produce antibodies. Now, I talked about vaccines. They spare us the symptoms of the primary response. Basically, they provide an antigenic terminant or an antigenic response without actually having the disease. And so they're immunogenic and reactive. They target only one type of helper, T cell, that's so fail to establish cellular immunological memory. That's what happens to some vaccines. So that's kind of important to know. It's interesting to know that it talks about T cells here, but the idea here is that it doesn't give us the long-term immunologic memory, but some immunizations, like vaccines. Okay, let's talk about vaccines. Flu vaccine. Flu vaccine only is very superficially, if you get a new one every year. Immunizations, like a tetanus immunization or a anthrax immunization, they don't just work on the T cell, they work on the B cell creating those memory cells, so it's a little different. Then we have passive humoral immunity. We talked about active humoral immunity before. We talked about that being B cell response. We talked about naturally acquired, artificially acquired, and passive immunity can be naturally or artificially also. Same thing runs here. Passive immunity means antibodies got into your body, but you did not create your own. How does that occur naturally? Well, for a fetus, via the placenta or via infant milk, milk through mom, the baby acquires antibodies from the mom that help defend it. The baby doesn't make any antibodies, so that's not an active process. It's passively providing the stage for the antibodies from mom to fight the infection. And so those are antibodies that are delivered to the fetus via the placenta or through the infant milk. You can also have it artificially acquired. That is, you have the disease, but you can't produce the antibodies. So somebody takes blood or serum from somebody who had the disease, and they take their antibodies and they put them inside of you. You don't produce any antibodies because you're borrowing somebody else's antibodies. And so the protection is immediate, but it ends when the antibodies go away. So the passive process means you didn't create antibodies. The active process is you created antibodies. The naturally acquired process for passive is you had to get through mother's milk or through the blood supply through the placenta. Artificial is always something that is injected into your body. If it's active, if a vaccination or immunization is injected to your body, which creates a low-level infection in you, of which your body is capable of fighting. On the other hand, in a passive artificially acquired immunity, there's just an injection of antibodies from somebody else that help to fight your battle. But you produce nothing. So let's talk about that in a different way. Humoral immunity. Active immunity. Naturally acquired means you got the infection. Passively acquired means the vaccine of dead or attenuated pathogens was put into your body. You developed a low-level infection, which allowed you to respond, which would allow you to respond if you got the infection again. The passive response is different. Their naturally acquired antibodies come from mom or from the milk or from the placenta. Artificially acquired means they inject you with the antibodies from somebody else who had the active disease. They came from here, and now you have them. But you produce no antibodies here, you produce antibodies here. Antibodies are immunoglobulins. There's a gamma globulin portion of blood, and they're proteins that are secreted by the plasma cells, those B lymphocytes, if you will, that attack the antigens that are foreign to us. They're capable of binding specifically with the antigen which was detected by that original B cell when it was named, when it first came in contact with it. So let's talk about a basic antibody structure, because the antibody, different kinds of antibodies, and they have different structures. They can be a T or a Y-shaped monomer, shape we'll show you in a minute, of 4 looped or linked polypeptide chains. They have 2 identical heavy H chains and 2 identical light chains. And there's a variable V region of each arm, which combines to form 2 identical antigen binding sites. And I'm going to show you that in a graphic in just a minute. And there's a constant C region of a stem, if you will, the bottom of each basic antibody, which determines its class. The antibody class could be IgM, IgA, IgD, IgG, or IgE, and they all have different locations of different things. They are activated by different antigens, or activated differently by different antigens. The cells and chemicals that the antibody can bind to is determined by that constant C region, and how the antibody class functions in an antigen elimination is again based on that C region. So let's run through that real quickly again. There are T or Y-shaped monomers of 4-loop linked polypeptide chains. Okay, so back to the biochemistry we talked about. There are 2 heavy chains and 2 identical light chains, and there are variable regions of each arm which combine to form 2 identical antigen binding sites. This is where the rubber meets the road, or the antibody can attach to the antigen. It's a very specific area because it can only hit whatever the antigen binding sites are has to be specific for the antigens that was developed during activation. There's a constant C region of the stem, if you will, of the antibody, which identifies the class, IgM, IgA, IgD, IgG, or IgE. And then it also helps to determine the cells and chemicals that the antibody can bind to, and how the antibody class functions as far as elimination of the antigen itself. So these all kind of work a little differently. This is a nice graphic of that antigen binding site. You see a heavy chain here, a light chain here. You see a little hinge region and a stem region. And the stem region is that the constant region. That's the place that's consistent, which identifies the class of that antibody. We talk about classes of antibodies, we talk about IgM. It's a pentamer. That means the first antibody is released. Or a pentamer, you'll hear both terms. It's a potent agglutinating agent, causes agglutination of the cells attached. It kind of causes foreign bacteria to be attached to each other, and it readily affixes and activates complement. So it's known for that. It's a shorter-lived antibody, but that's what it does. IgA is a secretory antibody, or secretory IgA. It's a monomer or a dimer. It's in the mucus and other secretions that helps prevent entry of pathogens. So you find IgA in your tears, in the mucus of your every orifice that comes into your body. This looks at that. This is the pentamer. It's a see that has 5 parts, penta, which attach which are kind of in this big long snowflake affair. And you get an idea because IgA exists in a monomer and pentamer. Pentamer formation, so it'll be in each one of these by itself or in the other, and there's the stem of each one. You get the idea. The monomer, which is attached to the B cell surface, serves as an antigen receptor. The pentamer, or pentamer, illustrated, circulates in the blood plasma and is the first Ig class released by the plasma cells during the primary response. This, in fact, is diagnostically useful because the presence of IgM in the plasma is very important. So it's kind of important to know that. And then IgM, its numerous antigen binding sites make IgM a potent agglutinator agent and readily fixes and activates complement. The IgA you see here, you see an IgA monomer which exists in limited amounts in plasma. The dimer, which is illustrated, the single unit, is referred to as secretory IgA, and it's located in saliva, sweat, intestinal juice, and milk. Helps prevent the attachment of pathogens to the epithelial cell surface, including mucous membranes and epidermis. So it has a little lesser role, as it were. We also have IgD and IgG. IgD is a monomer which attaches to the surface of B cells, and it functions as a B cell receptor in some cases. IgM is a monomer. 75 to 85% antibodies in the plasma are IgG. Comes from a secondary and late primary response. So usually when there's a secondary response, a delayed response, or memory cells are created, that's an IgG phenomenon. The first antibodies are created in the first response, the primary response, are generally IgM. So there's a high IgM response initially, but in the secondary exposure, it tends to be more IgG. And it crosses the placental barrier. So IgG antibodies cross the placental barrier. That's very important. And we'll talk more about that when we talk about the reproductive system. We'll talk about how IgG antibodies can be destructive to the baby during that. We also have IgE antibodies. They are monomers that activate some allergies and when parasites get into our body, or parasitic infections. They cause mast cells and basophils to break out those granules they have containing histamine and release it, creating an allergic response. And so if you were to take the blood sample from somebody who had a parasite, you would find they had lots and lots of these mast cells and basophils and eosinophils, so they're the same thing, that help these histamine granules. And there you see them. You see the IgD, IgG, and IgE, pretty similar in what they look like. And you see that IgD is virtually always attached to the external surface of B cell. IgG is that most abundant and diverse antibody in infection, and it presents 70 to 85% of the circulating antibodies in the body. And it protects us long term. So memory cells are generally IgG producers, which is kind of important. Remember that IgE is really an antibody that responds to allergic reactions and parasitic infestations. So we get a parasite or we get allergies, we tend to get a high IgE response. Talking about generating antibody diversity, there are billions of antibodies which can result from somatic recombination of the gene segments, if you will. There's hypervariability, lots of variable regions on some genes which increase the antibody variation through what's called somatic mutation. That's where each plasma cell can switch the type of H chain produced, heavy chain produced, making an antibody of a different class. And so that's just what you got. And this is a variation, if you will, of the theme. Antibody targets. Antibodies inactivate and tag antigens, period, and form antigen-antibody complexes, sometimes called immune complexes. Their defensive mechanisms used by antibodies include neutralization and agglutination. That's the two most important. They either neutralize the antigen or they cause the antigen to clump up. And then when they use precipitation and complement fixation, we'll talk a little bit about what those are in the next few slides. Neutralization is the simplest mechanism the antibody uses. The antibody blocks specific sites in the virus of the bacterial toxin, which prevents these antigens from binding to the receptors on the tissue cells. So the antigen is still there, but it can't do anything. The antigen-antibody complexes undergo phagocytosis, so it attaches to their antigens, which sort of alerts the phagocytes to go ahead and eat them. Agglutination is where the antibodies bind the same determinant on more than one cell-bound antigen, so they sort of clump all together. They bind to one antibody will bind to will bind to an antigen, and then the antibodies will bind to each other, creating a sort of clumping of the bacteria, which makes them sort of unuseful, or they can't function. Cross-linked antigen-antibody complex is agglutinate. An example of that is clumping of mismatched blood cells. That's where you put type A into type B people, and the antibodies will agglutinate the foreign blood type. There's precipitation, where soluble molecules are cross-linked. That's where the complexes precipitate and are subject to phagocytosis. So it causes the cells to precipitate this complement, if you will, and then what happens is it gets eaten by the phagocytes, the macrophages and neutrophils. Complement fixation and activation. The main antibody defenses against cellular antigens is complement fixation and activation. Several antibodies bind close together on a cellular antigen, and their complement binding sites trigger complement fixation into the cell surface. The complement triggers what's called cell lysis, or drilling those holes we talked about, and then activated complement functions by amplifying the inflammatory response, opsonization, and enlists more and more defensive elements. It activates more and more defensive elements to help take care of, if you will, or eliminate the foreign body. This talks about all of that process right here. What you're seeing here is really a graphic of the various responses that the adaptive defenses humoral immunity, that the antigen-antibody complex produces. Neutralization, agglutination, precipitation, development of complement, if you will, and ultimately phagocytosis, and the inflammatory response enhances that, the goal being to kill the cell, basically, as it goes. So the idea is the cell dies eventually, and it goes to those four mechanisms, or those 1, 2, 3, 4 mechanisms, which ultimately develop into that inflammatory response, which enhances the phagocytosis, which enhances that cell lysis. We have monoclonal antibodies, which are commercially prepared pure antibodies, and they're produced by what are called hybridomas. They're cell hybrids. They're a fusion of a tumor cell and a B cell. And they proliferate indefinitely, have the ability to produce a single type of antibody. These are used in research and clinical testing and cancer treatment. But wouldn't it be interesting to know that someday we get vaccinated against hopefully, against cancer? We have a B cell that's been activated against potential cancer cells, the type of cancers, if you will, that can attack us. That's where they're working right now, and that's a lot of work's being done in developing monoclonal antibodies. So far, we've talked mostly about B cells, but we need to obviously talk about cell-mediated immune response, and that would be the T cells. T cells provide our defense against intracellular antigens, and so we're talking more about that as we go. And we're really talking about virus infections, or viroid infections, or virions, or whatever, anything that enters the cell and causes a change. There are two types of surface receptors on a T cell. There are two types of surface receptors on a T cell, I'm sorry. There's a T cell antigen receptor, and there's a cell differentiation glycoprotein. That could be a CD4 or CD8 that's complement CD4 or CD8. And it plays a role in T cell interactions with other cells. So there are two types of surface antigens, that T cell antigen receptor, and then that cell differentiation glycoprotein, which can be made up of a CD4 or CD8. There are major types of C cells. That CD4 cell becomes what's commonly known as a TH cell, or a helper cell. And then there's the CD8 cells become cytotoxic cells, or TC cells, that destroy cells harboring any foreign antigens. Other types of T cells include regulatory T cells, or Tregs, and then memory T cells that work similarly to the memory cells, memory cells that go along with the B lymphocytes we talked about earlier. What you're seeing here is a graphic which explains the diversification of a T cell. We have a T cell, which is a T, which becomes it becomes a CD4 cell, a helper cell, or we have a T cell which is a CD8, which becomes a cytotoxic cell. What happens is the T cell, it goes to maturation. You see that CD4 cell here and CD8 cell here. When the CD4 cell comes in contact with that foreign antibody, it's activated, and it starts to produce again all these helper cells that down the pike, if you will. A TC cell goes through the same activation process, which is activated by CD8. It produces all of these other cells again that become cytotoxic cells. Both of these produce a memory cell, which allows that reaction to be much more effective and much quicker on the second, third, and subsequent exposures. The comparison of a humoral and cell-mediated response. Antibodies are the humoral response, the simplest ammunition of the immune response. So antibodies are very specific. The targets are bacteria and molecules in the extracellular environment, body secretions, tissue fluids, blood, and lymph. T cells of the cell-mediated response respond only to processed fragments of antigens displayed on the surface of body cells. Their targets are body cells infected by viruses or bacteria, abnormal or cancer cells, or cells that are infused or transplanted from somebody else. So when somebody gets a transplant, it's these cells that can actually mess up the transplant and cause a rejection based on that specific reaction. Antigen recognition, immunocompetent T cells are activated when their surface receptors bind to a recognized antigen that does not part of our self, not an id reaction. Id antigen. The T cells must simultaneously recognize non-self antigens and self antigens. So they have to be able to simultaneously be able to do both, because you want them to attack the foreign substance, not us. And so they have to be able to do that. It's a little more complicated. Those MHC proteins, there are two types of MHC proteins that are important to T cell activation, class 1 and class 2. Class 1 are displayed by all cells except for red blood cells, and class 2 are displayed by APCs, and that would be dendritic cells, macrophages, and B cells. Both types are synthesized at the endoplasmic reticulum, and they bind to peptide fragments. So there are two types of proteins that are important to T cell activation, class 1 and class 2. Class 1 is those proteins that are displayed by all cells. Every cell has those proteins. And then class 2 proteins were displayed by APCs. Those are like dendritic cells, macrophages, B cells. And so both types, again, are synthesized in the endoplasmic reticulum of a cell, and they bind to those peptide fragments. Class 1 MHC proteins. They bind with the fragment of a protein synthesized in the cell. It's called an endogenous antigen. The endogenous antigen is a self-antigen in a normal cell and a non-self antigen in an infected or abnormal cell. So here's what happens. The reason the antigen changes is because the cell becomes affected. It changes that antigen such that it becomes a non-self or a non-id antigen, and it shows the T cell that it's no longer one of us. It informs cytotoxic T cells the presence of a microorganism hiding in the cell. So cytotoxic T cells ignore the displayed self-antigens and go ahead and attack the cell. So here what you see endogenous antigen is degraded by protease, and so it's broken down. The endogenous antigen peptides enter the endoplasmic reticulum via the transport proteins. The endogenous antigen peptide is loaded into class 1 MHC protein. It said loaded MHC protein migrates to a vesicle in the plasma membrane where it displays the antigenic peptide to the outside world and ultimately to the T cell. And the T cell then knows that there's something abnormal going on inside of that cell proper. Class II MHC proteins bind with fragments of exogenous antigens that have been engulfed or broken down in a phagolysosome, or phagolysosome. They're also recognized by T helpers, so they bind with fragments of the antigens that have been engulfed or broken down by the phagolysosome. That means that once that phagocytic cell has destroyed that whatever is destroyed, and it extrudes that through exocytosis, that material, that protein binds to that and helps it be recognized by the T cell. Again, same process. Is the class II MHC is synthesized in the endoplasmic reticulum by an infected cell. The extracellular antigen bacterium is phagocytized. Could happen that way. The class II MHC is exported from the endoplasmic reticulum into a vesicle. You see that there. The phagosome merges with the lysosome, forming a phagolysosome antigen, it goes through a degradation process. The vesicle fuses with a phagolysosome, and an variant chain is removed, and the antigen is loaded. So this is now a different antigen than what is normal for that cell. What happens is a vesicle with the loaded MHC migrates to the plasma membrane and shows this peptide, if you will, on this class II MHC protein, which tells the T cells not normal. What's going on inside the cell is not normal. Okay, sorry, gotta hold that. T cell activation, APCs, most often the dendritic cell, migrate to lymph nodes and other lymphoid tissues to present their antigens to the T cells. The T cell activation is a two-step process: antigen binding, co-stimulation. Antigen binding is where CD4 and CD8 cells, that is the T helper cells and the cytotoxic cells, bind to different classes of MHC proteins, MHC restriction. The CD4 cell binds to antigen linked to class II MHC proteins of the APCs, and CD8 cells are activated by antigen fragments linked to class I MHC of APCs. So they do a little bit different activation. The dendritic cells are able to obtain other cells' endogenous antigens by engulfing dying virus-infected or tumor cells, importing antigens through temporary gap junctions with infected cells. And the dendritic cells then display these endogenous antigens on both class I and class II MHC proteins. T cell, the TCR then recognizes the non-self complex is linked to multiple intracellular signaling pathways. Other T cell surface proteins are involved in antigen binding, which include CD4 and CD8, which help maintain coupling during the antigen recognition. The antigen binding stimulates the T cell, but the co-stimulation is really important and is required before proliferation of the T cell can occur. And this takes you through that process where the dendritic cell engulfs an endogenous antigen, processes it, and displays its fragments in that type II antigen or type II protein we talked about. The immunocompetent CD4 cell recognizes that antigen and complex, and both the TCR and the CD4 protein bind to the antigen MHC complex. The CD4s are now activated, proliferating clones, if you will, and become memory and effector cells. So you see you get a memory cell and all these other helper cells in this case, because it's a CD4. We talk about co-stimulation of a T cell activation. It requires T cell binding to other surface receptors on an APC. Dendritic cells and macrophages produce surface B7 proteins when innate defenses are mobilized. So B7 binds with the CD28, that complement 28 receptor, on a T cell, and it's crucial to the co-stimulatory signal. Cytokines. Cytokines are cell killers, cell poisons, which include interleukin 1 and 2 from APCs, or T cells, which trigger proliferation and differentiation of activated T cells. So that's that co-stimulation step we talked about before. Without co-stimulation, energy occurs. Nothing happens. T cells become tolerant of the antigen, are unable to divide, and don't secrete cytokines. So co-stimulation is very important for these steps to not occur. Through co-stimulation, T cells are activated, enlarge, proliferate, and form those clones we talked about, which would be those activated cells as well as the memory cells. They differentiate and perform functions according to their T cell classes, whether that's a helper cell or a cytokinin cell. Primary T cell response peaks in about a week. T cell apoptosis occurs between days 7 and 30, so it starts to go through cell suicide. Effector activity wanes as the amount of antigen starts to decline. So as the antigen starts to go away, the T cell response goes away too. The benefit of apoptosis is activated T cells can be a hazard because they can start to attack other parts of our body. And memory T cells remain and mediate that secondary response in the future. So we don't want a bunch of activated T cells going around when there's no antigen for them to attack, because that could create lots of problems. And so they go through a self-destruct. The cytokines are mediated by cell development, differentiation, responses in the immune system. They include those interleukins that we talked about, and interferons. Interleukin-1, IL-1, is released by macrophages, which co-stimulates bound T cells to release interleukin-2, IL-2, or synthesize more of the IL-2 receptors. So basically cytokines include interleukin and interferons, and there's a process that we just went through on how those are developed. IL-2 is a key growth factor. Activated on cells that release it, and other T cells. It encourages activated T cells to divide rapidly. It's used therapeutically to treat melanoma and kidney cancers. And other cytokines amplify and regulate the innate and adaptive responses our body has. So they are synergistic to our body's innate response, that response that is not specific, and adaptive responses. The role of helper T cells, TH cells, play a central role in the adaptive immune response. Once primed by an APC presentation of an antigen, they help activate T and B cells. They induce T and B cells proliferation. They activate macrophages, recruit other immune cells. And without T helper cells, there's really no immune response. They're pretty important. Helper T cells interact directly with B cells, displaying antigen fragments bound to those MHC2 receptor proteins. They stimulate B cells to divide more rapidly and begin antibody production. B cells may be activated by helper cells, T helper cells, by binding to a T cell independent antigen. And most antigens require T helper co-stimulation to activate B cells. So T helper cells are very important for the process of the humoral response of the B cell. And this talks about that, where TH cells bind with the self-non-self complexes of a B cell that are encountered as antigen, and is displayed on the MHC2 of its surface. The TH cell releases interleukins and co-stimulatory signals to complete B cell activation. So you can see that process right here in that particular guide. Helper T cells cause dendritic cells to express a co-stimulatory molecule required for CD8 cell activation, the cytokine, the TC cell activation. This is a previously activated TH cell binds with the dendritic cell. The cell stimulates the dendritic cell to express co-stimulatory molecules, not shown, need to activate CD8 cells. The dendritic cell can now activate CD8 cells, that TC cell, with the help of interleukin-2 secreted by the TH cell. So let's tell the TC cell what to do. The roles of the TC cell are they directly attack and kill the other cells. Cytokinin is a cell killer. Activated TC cells circulate in the blood and lymphoid organs in search of body cells displaying those antigens that are non-self antigens, if you will. The role of the cytotoxic cells includes targets, which are virus-infected cells, cells with intracellular bacteria or parasites, cancer cells, or any foreign cell may be derived from a transfusion or a transplant. So you can see that in transplants, you'd be kind of a problem if you didn't make some account for the TC cell. TC cells bind to a non-self complex that can destroy all infected or abnormal cells. They provide a lethal hit, and that's where the TC cell releases perforins and granzymes by exocytosis. It releases those through its cell membrane. Perforins create pores to which granzymes enter the target cell, and granzymes stimulate cell suicide or apoptosis. In some cases, TC cells bind with the FAS receptor on the target cell and stimulate apoptosis that way. So what you're seeing here is a graphic which depicts that process. We can see that the TC cell binds tightly the target cell when it identifies a foreign antigen of MC1 protein on the target cell. The TC releases perforins and granzyme molecules from its granules by exocytosis. The perforins molecule inserts in the target cell membrane, if you will, polymerizes and forms a transmembrane pore, cylindrical hole, similar to those produced by complement activation we talked about before. Granzymes enter the target cell via the pores, and once inside, these proteases degrade the cellular contents and stimulate cell destruction or self-cell destruction. Say that fast 3 times. Natural killer cells recognize other signs of abnormality, and they lack the lack of a class 1 MHC, an antibody coating of a target, different surface markers for stressed cells. They see that and recognize those. They use the same key mechanisms as TC cells for killing their target cells. Regulatory T cells dampen the immune response by direct contact or by inhibitory cytokines. They're important in preventing autoimmune diseases. That means diseases where we start to destroy our cells, where we create antibodies or we start attacking our own id tissue, our own self-tissue. And this slide goes through those 2 processes. I'm not going to go through it in this lecture, but that's in your book, and you can take a look at that. A couple of variations we're going to talk about is really organ transplants. There are 4 types. There's autografts, isografts, allografts, and xenografts. Autografts are taken from one body site to another in the same person. For instance, a skin graft. Isografts are between identical twins. So if you have one who needs a kidney, you take the kidney from the identical twin. That's an isograft, and they'd be an exact match. Allografts are between individuals who are not identical twins, and then xenografts are from one animal species to another. Do we ever do that? Yes. We actually used to use a lot. Valves, heart valves from pigs. We'd put pig valves in human hearts because we didn't have any prosthetic valves at that time. So xenografts most certainly do exist, and they're still used time to time. There's a projection process where the body would attack it because it would be foreign. It depends on the similarity of the tissues. The patient is treated with immunosuppressive drugs to help reduce their ability to respond. Of course, many of these have side effects which prevent our ability to be immune competent to other kinds of infections. Corticosteroid drugs to suppress inflammation, anti-proliferative drugs, and immunosuppressive drugs which prevent the body from attacking that now transplanted tissue. Many of these have some very severe side effects, and they're significant. Immunodeficiencies, congenital and acquired conditions that cause immune cells or phagocytes or complement to behave abnormally or to not work functionally. So immunodeficiencies are where the immune cells, phagocytes, or complement aren't able to do what they would do in a normal person. B cells don't work well, T cells don't work well, if you will, not quite doing what it needs. There are also severe combined immunodeficiency syndrome. We'll talk a little bit about that. These are really caused by a genetic defect. Something happened that in the fetus where the normal scheme of things did not allow the fetus to develop its immune system. There's a marked deficit in B and or T cells. Abnormalities include abnormalities in the interleukin receptors, There's a defective adenosine deaminase enzyme, that would be ADA. Metabolites which are lethal to the T cells accumulate, so T cells start to eliminate. SCID is fatal if untreated, and its treatment is with bone marrow transplant. So it's really important that it's identified early, but it's genetic. The thing I want you to leave with is genetic in cause. It causes a detrimental effect on B and T cells. It causes abnormalities in the interleukin receptors. It causes a defective adenosine deaminase enzyme to be created, which is lethal to the T cell specifically, and so it's not conducive to living, if you will. There's Hodgkin's disease, which is acquired immunodeficiency, which is cancer of the B cells. It leads to immunodeficiency by depressing lymph node cells. So Hodgkin's disease is a type of cancer that affects the lymph cells directly, B cells primarily. There's acquired immunodeficiency syndrome, AIDS, which cripples the immune system by interfering with the activity of helper cells, helper T cells, so TH cells. It's characterized by severe weight loss, night sweats, swollen lymph nodes. Its opportunistic infection occurs where if you have this, it reduces, the AIDS reduces your ability to fight other infections, and so you can have other infections occur, which are called opportunistic infections. That includes pneumocystis, pneumonia, and Kaposi sarcoma, which is a type of cancer. It's caused by human immunodeficiency virus, HIV, which is transmitted via body fluids, blood, semen, and vaginal secretions. HIV enters the body via blood transfusions, blood-contaminated needles, or sexual intercourse and oral sex. HIV destroys the TH cells and depresses our cell-mediated immunity and that co-stimulation we talked about before. HIV multiplies in lymph nodes throughout the asymptomatic period. The symptoms appear in a few months to 10 years. HIV-coded glycoprotein complexes attach to the CD4 receptor. HIV enters the cell and uses reverse transcriptase to produce DNA from viral RNA. The DNA copy of provirus directs the host cell to make viral RNA and proteins, enable the virus to reproduce. So like other viruses, it enters the cell, it reprograms the DNA to produce more HIV to be then extruded back into the host. HIV reverse transcriptase produces frequent transcription errors, and there's a high mutation rate resistance to drugs. So when you make a drug against one version of that, it may not work against the next version because it mutates quite readily. The treatments with antiviral drugs and reverse transcriptase inhibitors, AZT. They are protease inhibitors, sequinavar and ritonavir, and new fusion inhibitors that block HIV's entry into the helper T cells. Autoimmune diseases. Where the immune system loses its ability to distinguish between itself and foreign antigens or foreign substances. We produce autoantibodies against ourselves, and sensitized TC cells are then go to attack body tissues that are our tissues. Some examples of that are multiple sclerosis, myasthenia gravis, Graves' disease, type 1 diabetes mellitus, systemic lupus erythematosus, SLE, glomerulonephritis, and rheumatoid arthritis. All those are autoimmune diseases where we create antibodies against ourselves. The mechanisms of an autoimmune disease include foreign antigens may resemble self-antigens. The antibodies against the foreign antigen may cross-react with our own antigens, and new self-antigens may appear generated by gene mutations, changes in self-antigens, by hapten attachment, or as a result of infectious damage, or release of a novel self-antigens by trauma of a barrier, the blood-brain barrier, for instance. So there's three mechanisms which that can occur. We have hypersensitivities, immune responses to a perceived, otherwise harmless threat, which causes tissue damage. Their different types are distinguished by their time course or whether antibodies or T cells are involved. Antibodies cause immediate and subacute hypersensitivities, and T cells cause a delayed hypersensitivity. So hypersensitivity includes immediate hypersensitivity. Acute type 1 hypersensitivities are like allergies. They begin in seconds after contact with an allergen. If you have a reaction to a bee sting, you develop a type 1 reaction. You go into anaphylaxis, meaning that you stop breathing. Initial contact may be asymptomatic, but sensitizes a person. So the first bee sting may cause nothing in you. It's the subsequent bee stings that will cause that problem. Reaction may be local or systemic. A bee sting could make you stop breathing if you have anaphylaxis, or it could just cause a lot of edema and swelling in a local area. The mechanism involves the IL-4 secreted by T cells. IL-4 stimulates B cells to produce IgE, that antibody that occurs when we have an allergic reaction. IgE binds to mast cells and basophils, resulting in a flood of histamine throughout the body and inducing an inflammatory response. Anaphylactic shock is what I just talked about. It's a systemic response to an allergic reaction that directly enters the blood. Basophils and mast cells are enlisted to the body. Systemic histamine releases may cause a constriction of the bronchioles. We can't get air in, like an asthma attack. Sudden vasodilatation and fluid loss to the bloodstream, so fluid leaves our cardiovascular system and goes into the skin or the tissue. Hypotensive shock and death, and the treatment is with epinephrine, which works to block that effect. We have subacute hypersensitivities, which are caused by IgM and Ig transferred via blood plasma or serum. They're slow in onset, they can take 1 to 3 hours, and they have a long duration of 10 to 15 hours. There's cytotoxic type 2 reactions, and those are antibodies where the antibodies bind to antigens on specific body cells, stimulating phagocytosis and complement-mediated lysis of the cellular antigens. The example is mismatched blood transfusion. So if you put a type B into type A, a type B blood into type A, you get a type 2 reaction. We also have a type 3 reaction, which is a subacute hypersensitive reaction, which is for immune complex type 3 hypersensitivity. Antigens are widely distributed throughout the body or blood. Insoluble antigen-antibody complexes form. Complexes cannot be cleared from a particular area of the body, and intense inflammation, local cell lysis, and death result. Example is systemic lupus erythematosus, SLE. SLE is really an autoimmune disease of your body. It attacks all your organs. We have delayed hypersensitivities, which is called type 4. It's slow onset. It can take 1 to 3 days. The mechanism depends on helper T cells or Th cells. The cytokine activated macrophages and cytotoxic T cells cause the damage. An example is in allergic contact dermatitis, like in poison ivy. Alrighty. We have just gone through the recorded lecture of the immune system, Chapter 20, for the A&P 230 second semester class. Thank you very much.